Hey, Hammy here, back with part 3 of section 7.1, in which we're going to look at finally the discovery of DNA part 2, when we finally get to the actual kind of figure out the structure of this DNA molecule. Before we actually get to Watson and Crick, there are a few people leading up to their discovery that contributed to how they kind of figured their final result out of, the final structure of DNA out. In the mid 1900s, there was Erwin Chargaff. Okay, Chargaff was an Australian biochemist. And he did analysis on a bunch of different species, and they knew that DNA contained 4 different bases, okay, nitrogenous bases. And they knew that these nitrogenous bases consisted of adenine and guanine. We usually just use an A for adenine and a G for guanine. And they had kind of a double ring structure that they called purines. And there were 2 single ring structures, cytosine, which we usually use a C, and thymine, which we use a T, which were single ringed nitrogenous bases that we call pyrimidines. And scientists knew through chemical testing that there was this A, G, T, and C. There were 4 different nitrogenous bases in DNA. And when Chargaff started testing different creatures, Streptococcus, which is a type of bacteria, prokaryote yeast, which is a eukaryote, a herring or fish, and a human, he noticed something. Well, he noticed 2 things. First off, he noticed that the numbers were not the same. The concentration of the 4 bases differs from one species to the next. Okay, different species had different amounts of A, T, G, and C in their DNA. The second thing he noticed, and we now call this Chargaffs Rule, is that within each species, if you look, let's go to the human here, 30.9 and 29.4. So they're both about 30%. And then between G and C, we're both about 20%. Okay, so 30 plus 30 plus 20 plus 20. Okay, that would equal then your 100%. He noticed that there always seemed to be about the same amount of A, or adenine, as there was thymine, or T. And there was always about the same amount of guanine as there was cytosine. Okay, and he didn't quite know what to make of this other than he found this out and he published these results. When Watson and Crick would see this, that would be critical in kind of putting together that final structure of DNA. Another very important person in the discovery of DNA was Rosalind Franklin. And she's finally starting to get a little bit more credit where credit is due. It was kind of ignored for a long time. The background story to this is quite interesting, and we'll probably kind of review this a little bit in class. But it was hard in the early 1900s to be a female scientist, and she had to really fight for her research. And one of the things that she did was she would take DNA and crystallize it and take a picture of it with X-rays. And they were doing this at the time with different kinds of biomolecules. And depending on that X-ray would show up, it would give them an idea as to the atomic molecular structure of different compounds. Well, when she took an X-ray diffraction of DNA samples, she got this iconic kind of X pattern here. Okay. This X pattern gave them different measurements and also told them that DNA was kind of a helical pattern or double helix, where there were 2 strands wound around each other. And again, this was very helpful for Watson and Crick when they were coming up with their final structure. Okay, James Watson was an American biologist who actually went over to England to work with Francis Crick, okay, at the universities over there, who was a British physicist. And as they began working, they sort of began talking, and this idea of DNA really began to fascinate them. And there were several people in the mid 1900s that were really working to figure this out. One of the most famous was Linus Pauling, chemist in California, and different people in various universities were really scrambling once Hershey and Chase figured out that DNA was the genetic material. They really began to figure out, well, what is its structure and how does it work? They looked at, the Chargaffs rules and Rosalind Franklin's picture, and just what they knew about the different the components of DNA, and they kind of put together that DNA was made up of nucleotides. Okay, one nucleotide has three things. Okay, it has a phosphate group, and then this pentagon type shape down here, and it's missing the carbons here, but a 5-carbon sugar called deoxyribose sugar. Okay, and then hanging off of here, this is just one of the sort of the single rings here. This was the nitrogenous base up here, which we knew was A, T, G, or C. Okay, and they actually made cardboard cutouts of these things. The one video we'll watch in class actually shows taking these things and seeing how they kind of would lay them out on the table, and kind of knowing their chemical properties, tried to figure out how all these things fit together. When they finally had kind of an aha moment, that was in 1953, and they actually kind of figured out how this DNA works. Several people had put they figured out it was kind of a phosphate to sugar backbone down the sides. But like Linus Pauling had 3 strands with the sugar phosphate backbones in the center and the bases sticking out. Okay, and Watson and Crick kind of did the same thing, but when they talked to Rosalind Franklin about this, she actually laughed and said, death to the DNA, because this can't be right. But then they had an aha moment, and they said, what if we turn the bases inward? And what if, using Chargaffs rules, what if, to keep the spacing right, okay, the measurements from Rosalind Franklin's picture, the measurements worked out if they had a single-ringed purine connected to a double-ringed pyrimidine in the center, held together by a hydrogen bonds in the middle, represented by these dashed lines here, okay, down the middle of the molecule. And then the sugars and the phosphates, sugar, phosphate, sugar, phosphate, sugar, phosphate, kind of down the backbone. So those were the sides, kind of like a ladder. And then the nitrogenous bases were the rungs. And the whole thing, okay, was twisted into this double helix. And when they started looking at the size of the molecules and they started looking at these measurements, okay, they fit with what the X-ray diffraction data said from Rosalind Franklin. And so they actually built this thing with their cardboard cutouts and chicken wire, and glued this and built this thing from floor to ceiling. They also said that it is anti-parallel. So these 2 strands, you see the sugars, the oxygen end is pointing up over here. Okay, over here it's actually pointing down. They actually said in order for this to work, the 2 things have to run in opposite directions. Okay, so they're not running the same way, they're running in opposite directions. Okay, and so they're anti-parallel. And they published actually a short paper. You can Google search it. It's only about a page or 2 long, and it's where they kind of put forth their structure of the DNA, and they are widely known and given credit for finally figuring out the structure of DNA. And that led to just kind of an explosion of research on how exactly this DNA code makes a copy of itself, and how it's made into the proteins, and how these genetics are expressed, which we'll get into in future videos.